Okay, I think we can start with this event. Sorry for this technical suspense moment, but these things you cannot predict. Anyway, I think we are all there and we are ready to start with this final event of TRAIN project, which is the Unconference. We decided to choose for this format in order to facilitate the communication between all of us and the public potentially present at the final event. The initial idea was to have it in a public place where also citizens could have a direct feedback on the major achievement of the project and what could be the evolution of TRAIN. Unfortunately, we cannot, but we are happy to see that many participants are joining, not all related to project partners. So possibly we will have this opportunity anyway, and after the presentations, we will have the time for discussion. We also thought that we may record part of this event in order to create a final video that could be eventually distributed to have an overall idea of what TRAIN has been, who we are, what we did, and so on. So this is also what we are going to discuss today. As for the format of the Unconference, we didn't want to make everything clear in the agenda, so the details of the speakers, their presentation, what they will say is not there. It will be a kind of surprise. And in this spirit, we want to leave everybody free to choose what they believe is the most important perspectives and content, in their opinion, to foster discussion and possible future developments. I think that to start, we can just welcome everybody in the 3 languages as it should be done for this kind of Interreg project. So I will spend a few words in Italian as well, and then Bostian will just convey the welcome address in Slovenian as well. So, benvenuti a tutti a questo evento finale di TRAIN, una Unconference, per spiegare e presentare i risultati di questo progetto Interreg Italia-Slovenia che ha unito le competenze dei diversi partner e messe al servizio della biotecnologia e dell'innovazione. Lascio la parola al mio collega Bostian per il suo saluto in sloveno.
Pozdravljeni še z moje strani, slovenski nagovor. Hvala vsem za udeležbo na tem zaključnem dogodku TRAIN, ki bo predstavil ključne rezultate tega projekta iz področja biotehnologije. To bodo vključevali tako znanstvene kot podjetniške rezultate in upam, da boste ob tem uživali. Potekal bo nekoliko bolj sproščeno v formatu Unconference, to pomeni, da boste lahko udeleženci aktivno sodelovali pri tem. In na koncu bo tudi možno za diskusijo med udeleženci, tako da svetujem, da vsa vprašanja prihranite za takrat.
Thank you, Bostian. So obviously the rest of the conference will be in English in order to let everybody understanding and sharing ideas. And I think we can start with the first session, which is related to the TRAIN project itself. So myself, together with Bostian and Francesca from expert team, will present you the results of the project. So as you probably all know, TRAIN has been composed by different partners. Some are academics, and in this case, it's ourselves, the ICGP, the International Center for Genetic Engineering and Biotechnology, and myself. We are the project coordinators, and the other academic partner is Joseph Stefan Institute in Ljubljana, and Sasho is there and will also present what we have developed together. Together with the postgraduate school of the institute. And then TRAIN also team up with clusters of companies from both the Slovenian side and the Italian side. And the idea was to facilitate the transfer of technologies and research results to the industrial sector, and to create platforms where more and more researchers and entrepreneurs can meet, discuss future developments and so on. And so we pick up also one company from the Venice territory, expert team, and we try to develop together some translational project that we will show you today. So these are the main partners, and you will meet them individually during this unconference. So I won't spend too many words now in introducing everybody, because we can do it one by one when they will be with us and speak and present. The idea of TRAIN overall started when we decided to match the competencies that exist in our research institutes on biotechnology, the possibility to generate large amount of data using omic technologies and high throughput screenings. These technologies currently generate tons of data that needs to be stored and analyzed. And our colleagues at Joseph Stephen actually had the skills and competencies to analyze large amount of data that are generated by wet screenings, wet experiments, and to elaborate them and generate additional virtual analysis. So that was the initial idea that drove us to team up and to merge our competencies, and then to offer what we can produce to companies existing in our transporter territories. So with this in mind, we decided to undertake the project, and today we will hear what we could do and what we intend to do, starting from where we are now. So I think I more or less introduced the project and the partners. We will have some time for discussion at the end, and everybody is welcome to write if there are any questions in the chat. Bastian will collect all questions and we can have a final discussion at the end. So with that, I think I will leave the floor to Bastian from Technology Park Ljubljana, one of the partners from the Slovenian side, who will tell us what they did and what is the biotechnology field in Slovenia at present. Thank you, Bastian.
Yes, thank you, Serena.
I hope you can hear me now. Thank you, Serena. So I'm going to present some of our insights which we gathered from this project, which was very useful for us. First, I'm going to start actually with more a global picture. We live currently in 2020, and there are important milestones being reached also due to COVID. And some of the trends which are going on is a shift from the one-size-fits-all type of treatment for patients to a more personalized approach based on the characteristics of each individual patient. Then there is greater collaboration between companies, which means that companies are forming joint projects with each other. They are not just competition, but they are also collaborating with each other to launch new products on the market together. Then also biotech, as a very interdisciplinary field in itself, is also including a lot of other technologies, also the digital ones, for example, the cloud management, virtual reality, AI, and so on. And it is really helping with the processing of big data and stuff like that. Then also there is a trend of optimizing the regulatory procedures for drugs, because this takes a lot of time, and procedures are going on that would speed up this process and would enable drugs to be on the market faster. And lastly, also advances in important fields of genetics and immunology allow us to diagnose and treat more diseases. Okay, so moving on. In Slovenia, companies are also following these trends, mainly related to the digital technologies. In Train, in 2018, we identified 27 biotech companies. This means that they devote 75% of their budget towards R&D in biotech. And they mostly represent small and medium enterprises, which offer products or services to large companies abroad or at home. And these are large biopharma or medical companies. Or on the other hand, they conduct research through European grants or national grants or via public financing. So then we asked the companies what they lack the most, and we found out that actually they lacked networking opportunities and human capital. Of course, networking is very important for entrepreneurs because more opportunities to meet people presents more opportunities for future collaboration. And of course, human capital is very important in the long run for companies because it enables them to keep an edge in view of their competition. And this includes not just hiring high quality staff, but also training existing staff. And what we lastly also found out is that there is quite large fragmentation of the R&D sector. This means that the institutes and companies who are conducting research are not well connected with each other. They do research just by themselves and do not share it. So here the state should intervene and maybe support this process. So then our process next was to address these needs and help companies and also researchers in this field. And firstly, we organized headhunting events where companies could meet students, PhD researchers, or just generally people with ideas who would be interesting for the market and to connect them with each other. And these were quite successful. And actually on this Wednesday, we are organizing another one, and at least 40 students will join. So we hope it will be a successful one. Then we also organized workshops, which focused on different healthcare topics to educate companies and to make it easier for them to partake in the market. And also, lastly, a very important event is the Health Day Conference, which is a yearly event. This year, it will take place on 22nd of October, and you're also kindly invited. Each year, it focuses on another health topic. And it is meant to promote companies and new technologies in this field. We also developed some online tools. First one is the resource container, which actually contains all the materials from Train. And this serves for people and enterprises to gather information in Italy and Slovenia about how to enter the market more efficiently. And we also developed a concept for the biotech station. This is actually a platform which will connect the stakeholders in this area in the biotech. And we will organize events in the future to try to connect stakeholders, find investment opportunities for companies, also organized one-on-one meetings for companies and so on. So Train activities will definitely continue after the project ends. And lastly, we also, together with several other project partners, we developed 2 virtual reality simulations. This will be also presented in this event in more detail. The aim of this virtual reality simulation was to present the scientific processes which partners are undertaking in their institutes. And first one was devoted to drug development. So what kind of methods are used in the lab to discover new drugs? And the second one was how food bacteria are detected generally. So now we are going to show a video of one of the virtual reality simulations. And I kindly ask Serena to launch it. I will stop sharing. Okay. Okay, sorry for the technical difficulties. Just to give you an introduction for this VR simulation, I hope it will run smoothly because with this transfer via internet, maybe it will stutter a bit, so sorry about that. But generally, we will show just how drugs are developed in the lab, and but this process will be talk about more deeply also in the next speeches. So I will not focus very in detail here. So and David from the designer team who designed this simulation will talk. So I will just let it run now.
Of experience we've developed in cooperation with Technology Park of Ljubljana and Joseph Stefan Institute and the ICGEV of Trieste. So let's begin our tour. Here we are in a virtually recreated lab featuring some of the actual equipment from ICGEB. This equipment has been 3D scanned by us and represents a fine sort of a gamification addition to our experience. So we've included a lot of data. We've really aimed for the high fidelity representation of the work that's being done in the actual labs. Well, let's now begin the robotic procedure.
Just to stop here a bit, this process actually includes robotics, and this helps us to quickly discover new drugs, because in order for you to find one suitable drug, you have to test several thousand other ones, and using robots, this actually quickens the process.
And the tray goes to a PCR machine. Okay. That's visually pretty much the first room. So let's move on to. Welcome to the chip environment. Right now we are in silico. We are virtually inside a circuit board. And in this environment, we're going to take a look at how machine learning looks like in a virtual reality environment. This, I believe, is the first demonstration of this kind. So let's take the compound screen and insert it into our servers.
So now here we are going to teach the computer to find the right molecule for us. We are using live laboratory data from the experiment which we saw before. And then we will generate a computer model which will actually help us to identify future molecules which would be suitable for our use, for example, to treat heart disease.
And observe the creation of a random forest. A mathematical concept that we've aimed to recreate not only visually, not only three-dimensionally, but in virtual reality itself, which could be argued is the fourth dimension. So now that the random forest is being created, let's take a look Okay, the counter has reached 95%, so the quality of the random forest is now sufficient for the next phase.
Okay, so now we train the computer, and now the computer knows that he needs to search, for example, for molecules which would treat heart disease, which would cure it. So now we're going to go through a database of a public database which contains several thousand random molecules, and we will input this in the model, and then it will give us back the possible molecules which can be used for actually treating heart disease.
These tablets right here represent the molecule databases. Perhaps we should include one that's very popular in recent months. By the way, the remdesivir was created, well, his effects, his possible effects were discovered using a process not like this one. So now that we've got a random forest created, let's put the entire PubChem library through a random forest. Each molecule goes through the entire forest where the effects are analyzed. And when the molecule comes out, we'll have a far broader picture of its effects. Oh, look. Here are our molecules. The results are here. Now we know much more about each molecule. Let's not lose anyone. Come on, go to the server. Okay, we are really excited about this room. And here we have our select drug candidates. The three molecules that we presume, or a couple of molecules that are looking most promising as the drug candidates. Okay, this is the room most enjoyed by the younger audience who is yet to enter science. And the goal of this room with perfectly functioning physics is to find the correct molecule for each ailment or for its effect. So we've got 3 cubes with which we can interact with and observe the high fidelity images from the processes. So let's go about finding the right pair. No, this one is not it. How about you? Are you the right pair? Oh, no. Then it should be.
This process is sped up a bit. You just make the right molecules with the right effect.
Here we are in a hospital environment where our patient is awaiting his newly created treatment. When we get closer, he sort of disappears, so we can focus on his heart with which we can interact. So let's use the cures that have been developed. This one starts to regrow the blood vessels. You see, the formation of new blood vessels is increasing. And this one will heal the fibrotic tissue. You see, fibrotic tissue is disappearing. Now we just have to restore the proper heart cell count. The visible effect of this is the heart beating faster, well, normal, again. This has been the simulation. Thank you for your attention, and goodbye.
So thank you. These processes will be elaborated more in depth in next speeches. So I thank you for your attention at this point.
So thank you very much, Bostian. We were very happy about this virtual reality. Of course, we hoped to show it to everybody in real person. It is a very nice and funny experience. I can ensure that everybody who tried it did have a lot of fun. Maybe we can have the chance to do it in our next Interact if we will get funded. And I think this is also very nice to distribute and let people try to experience the screening, since now in COVID time we are talking about screenings, virtual screening for drug repurposing. This is kind of news that we hear every day. So it is very good, I guess, to let people having a real idea what the screening is, what the virtual screening is. And so it is very timely. And of course, also at ICGB, we have undertaken some screening for COVID. So maybe next virtual reality could be about that. All right, so I think we can move on with the last presentation in this session, who is Francesca Bruno from Expert Team, with whom we try to develop some new assay to detect the presence of pathogens in the environment, food, but not only. So please, Francesca, go ahead.
Morning, everybody. Can you hear me?
Yes, we can. Yes, we can.
Okay.
Yes.
Yes.
I can share my screen because there is... Okay, perfect. Okay, so during this project, Expert Team was focused searching pathogens in food because nowadays illness resulting from the consumption of food or water contaminated with pathogens and or their toxin have a wide range of economic and public health impact worldwide. It's a very big issue to recognize and detect food contamination during quality control of food before it's launched on the market. So the initial idea shared with ECGB was to test by a molecular method, so PCR, polymerase chain reaction, the presence or the absence of one or more pathogen in bacteria in food, and then verify that the pathogen is alive or dead by high content analysis using the facility that are present in ECGB. At the beginning, the idea was to test the presence of the pathogen by multiplex PCR developed by Expert Team, and then test by two different assays that were ERF3 GFP and EFN reported system, the presence of the live or dead pathogen. But test assays don't discriminate between pathogen and non-pathogens, and they are not sufficiently sensitive for our purpose, so we decide to quit this approach and to test another way. So foodborne pathogen detection is one of the most important problems when dealing with food, and to now the most important techniques to detect pathogen contamination are conventional microbiological methods and PCR or real-time PCR methods. Conventional microbiological methods rely primarily on plating, by direct plating and biochemical tests, but they are time consuming and also pure sensitive, while PCR methods or real time are more specific, sensitive, and with speed of detection and results. So we wanted to use PCR methods. But the problem with PCR methods is to discriminate between live and dead cell, because PCR methods are based on amplification of DNA that doesn't provide information related to ability, as DNA molecules from live that are the problematic ones and that pathogen can be distinguished by PCR. So when DNA detection methods are used for analysis of food, one of the major concerns is false positive results from the detection of dead cells. So as the distinction between viable and dead pathogen is important, we test a new approach based on mRNA analysis, because most mRNAs in living bacterial cells has an half-life of only a few minutes, while bacteria DNA is more stable and in target organism, when the organism is died. So the detection of mRNA can be a good indicator of viability of cells. So our new approach that is based on the detection of live microorganisms using TACMAN-based quantitative reverse real-time PCR that amplify mRNA of the pathogen. So the mRNA is reverse transcribed in cDNA, and then amplified. So one of the first pathogens that we studied was Salmonella enterica. The Salmonella enterica contamination in various food is a significant public health concern, domestically and internationally. For a successful detection of pathogen using mRNA, we selected the INVA gene, because it is present in all pathogenic serovars of Salmonella described to date, and the product of the gene is essential for the organism's ability to invade mammalian cells and to cause the disease. In order to determine the feasibility of the use of the INVA gene mRNA as a marker of viability of Salmonella, we test two different types of Salmonella samples and two different strains. The samples were dead, so lysed after being boiled at 90 degrees for 30 minutes, and live, lysed directly. So after lysing samples, mRNA was extracted, reverse transcribed in cDNA, and then amplified by qPCR, so the real-time PCR, with specific primers and probe, to assess the difference between live and dead pathogen. In each run, a reverse transcribed sample, so cDNA, and the corresponding RNA samples, so not reverse transcribed, were amplified. And if the difference between the threshold cycles between the cDNA and mRNA were greater, so non-reverse transcribed, were greater than 4 cycles, we consider samples positive for the presence of the microorganism. While in the case of CT values that dipped below this range, so the difference between the two cDNA and mRNA is below 4, the sample is considered negative for the presence of live and active microorganism. So the other pathogen we studied was enterohemorrhagic Escherichia coli that caused foodborne illness worldwide. So not all the genes were suitable targets for this kind of pathogen, and our work regarding Escherichia coli was based on RFB genes that encodes for the lipopolysaccharides O157 and is unique for this serogroup. This gene has been identified as a good viability marker because it is transcribed in all growth phase from early exponential to late stationary phase. And so this mRNA is present only in live cells and degrades very quickly after cell death. So the same work was made with Salmonella, was made also with Escherichia coli. Samples and dead samples were treated as in Salmonella experiments. So reverse transcribed samples, cDNA, and the corresponding mRNA were amplified for each experiment, and samples containing live Escherichia coli cells were considered positive, as the difference between the CT values for qRT-PCR and qPCR is greater than 4 cycles. So samples including dead Escherichia coli were considered negative for the presence of live cells, observing that the difference between CT values of reverse transcribed samples and non-reverse transcribed is similar. So at the end, once single real-time PCR were tested, we developed a unique multiplex qPCR that in a single reaction in the same mix is able to detect 2 different pathogens, Salmonella enterica and Escherichia coli. We ran in a unique assay to test multiplex efficiency to detect Salmonella and Escherichia coli, in which the live cDNA of both pathogens was amplified, were put together in the same well. In both rounds of this experiment, the mRNA of both pathogens was amplified successfully, and our pilot action to develop an assay distinguish between live and dead Salmonella enterica and Escherichia coli was successful. So with this developed method, we are able to detect foodborne pathogens, and the most important, to discriminate between live and dead pathogen. So regarding this part of the project, we decided to develop virtual reality simulation in collaboration with our partners. And this activity can be useful to disseminate results of the project and to make scientific reality known in a simplified but direct way to which whoever a scientific background and for those approaches to these topics for the first time. So thank you for your attention.
Thank you very much, Francesca. Also, to keep the time, yes, I want to remember all speakers that, of course, it is very important that we stick to our 10 minutes in order to let everybody having the possibility to share his her opinion. So we can move to the more technical session. First speaker will be Sasha Zarowski, who is the head of the laboratory with whom we partner for this for the academic part of our Interreg. So Sasha, please go ahead. Unmute your microphone.
Oh, the host was kind to unmute me. Thank you.
Right.
So my name is Sasha Zarowski, and I am now actually head of the Department of Knowledge Technologies and the Jožef Stefan Institute in Ljubljana. The team for the joint Interreg project include Dragi Kocev, Panče Panov, Tomaš Tepišnik, and Martin Breskvar. And we were actually dealing with the task of applying different machine learning approaches to analyze data from high content screens with the purpose of drug development and repurposing. And we analyzed different kinds of high content screens, including compound screens, where you apply different chemical compounds from compound libraries to a cellular assay, but also genomic screens where different components of the genomes are interfered with and the effect of this is assessed. So in high content screens, it's typical that you have images of the cellular assays taken under the microscope, and then we use machine learning to learn models that relate on one hand the compounds, or on the other hand the microRNAs, and on the other hand the effects that these have on the cellular assay, which are typically some features extracted from the images. And once we learn such models, we can apply them for virtual compound screening to identify novel drug candidates. And this was very nicely shown in the virtual reality presentation. So in analyzing data from compound screens, we are really learning structure-activity relationships. We are learning to relate the structure of the compounds, which is shown here on the left-hand side, to their activity, which is represented here as a number. And in our case, typically, we have not only one target activity that we are looking at, but we have several features like intensity of collagen and alpha SMA in the particular screen of fibrosis that was addressed. So we get the compounds on one hand, and we have the effects on the other hand. And to this, we apply machine learning methods. I cannot go into the details of this, but you saw in the virtual reality representation, there were random forests. The random forests consist of trees. And here you can see in the trees that you have different features. So this tree is actually from analyzing a microRNA screen, and what is in the nodes of the tree are different functions or annotations of the genes that are targeted by the miRNAs. So this is just to show you that these trees actually also have some meaning and can be analyzed, and some interesting knowledge can be gained from the trees. But to get good predictive models, we are typically actually using a number of such trees, and the number of trees, these are called forests because there are many trees in the forest. Okay, and once we use the data from the compound screen as training data, we apply machine learning and we get this model for virtual screening. Then we apply it to lots of compounds which are potential drug candidates to get predictions for these drug compounds. And this allows us to be much more efficient in identifying new drug candidates, because the physical tests in the laboratory, of course, they cost money and they cost time and labor, whereas applying the virtual screening models that are learned with machine learning, we can check many, many more compounds. We can check millions of compounds very, very easily. And so the compounds are typically described by some structural features, which are called fingerprints. And on one hand, we have these fingerprints that describe the compounds. On the other hand, from the photographs taken under the microscope, some features are extracted, and these are then the targets that we want to predict by using machine learning. So the particular screen that we actually analyzed, the first one, was concerned with reducing fibrosis in myocardial infarction. So this is to help the heart regenerate after heart attack. And the features extracted from the images were the intensity of alpha SMA and collagen, and the compounds were described by the structural fingerprints that I mentioned. And so with these models learned, we actually conducted virtual compound screening in this PubChem library, which was also shown in the virtual reality. Here we had close collaboration with Nadia and Serena that actually checked closely the predictions of the virtual screening model, and then actually tested in the wet lab several candidates, of which one was found to work really, really well. So actually, within the project, we analyzed several different screens, I believe 3 different ones, including compound screens and microRNA screens. And we actually came to the conclusion that we can also use models learned from microRNA screens to perform virtual compound screening. And this is very useful. I have to say we had excellent collaboration with ICJEB. This was a great pleasure to collaborate with them. And also actually the experience of creating the virtual reality was quite interesting. And I hope you like the virtual reality representation of this whole process of virtual compound screening. It is a relatively complicated process, but the virtual reality gives some insight. Thank you very much for your attention.
Thank you, Sasha. We confirm that we had agreed to time and occasions to really merge what we can do and what you could do, and to learn a lot reciprocally. So Now I will give the floor to Nadia, who will quickly present our side of this collaborative work. Thank you, Nadia.
Hi, everyone. I'm going to show you quickly the science behind the VR. So we've talked a little bit about the, there we go. We've talked about the virtual reality demonstration that Tavarna made for us. And you've seen a little bit about what Sasho at JSI did with the data. And I want to show you what it looks like from our side. So this is the lab that you saw, the facility, the screening facility from the virtual reality standpoint. And you have a computer screen and you click on the computer screen and it says the screening has started and the robots start to move. But from our side, it looks a little bit different. And to begin, I wanted to explain to you what is a screening. So a high throughput screening is an area of expertise in ICGB and was also a focal point of this interregional project of TRAIN. So in a screen, to simplify it, you have a scientific problem. You simplify your problem by building a simple model of your problem. Then you add a collection of molecules to this model in order to hopefully find a solution. So in our case specifically, we're looking at the issue of a lack of cardiac regeneration after a heart attack. The heart doesn't function very well anymore after a heart attack, and you break it into a lack of vascularization, so blood vessels don't form, a reduction of contractive tissue, and a formation of scar tissue. This can be simplified some more into lack of vascularization can be shown by endothelial cells, which are unable to form tubes. Reduction of contractive tissue is a lack of cardiomyocytes proliferating, and the formation of scar tissue is due to fibroblast activation. Then we build these three models and we add either a library, which is a large collection of chemical compounds, or a library of RNAs. And hopefully we identify compounds or RNAs which can increase proliferation of endothelial cells and cardiomyocytes, or reduce activation of fibroblasts. The solution is unbiased because before we start, we don't have an idea of what we're going to find. And it's also based on function, which is different than other approaches where you're just looking at what a cell is doing. In this case, you're looking in this model, which molecules really have an effect on my model and can solve my scientific problem. So as Sasha also mentioned, in this case, we were focused on fibroblast activation. So unlike the virtual reality where we just have to click on a computer screen and we start a screen, in this case, we have to start with culturing the cells. And for sure, you've seen this very often now, these beautiful images that are on all the posters. So these are the fibroblasts that we investigated in our screening. Fibroblasts are cells that are found in connective tissue. And if you have an insult or an injury, such as a myocardial infarction, a heart attack, you have molecules that are activated, such as TGF-beta, which cause these fibroblasts to turn into myofibroblasts. These myofibroblasts are larger and they produce more contractile tissue, more contractile protein, such as alpha SMA, and they produce more connective proteins, such as collagen. The alpha SMA promoter will activate alpha SMA. The collagen promoter will produce the production of collagen, but these are invisible. So in this case, we used fibroblasts from the heart that were genetically modified. So we have the alpha SMA promoter. It doesn't only produce alpha SMA, but it also produces the red fluorescent protein. Our collagen promoter doesn't just produce collagen, but also the green fluorescent protein. And that gives us these cells that are green, red, or a mix of green and red. So then this is our in vitro model of fibroblast activation. So we have fibroblasts from the heart, which are already green because fibroblasts generally express collagen. But as soon as you add TGF-beta, the cells are much more green and much more red and have a phenotype of myofibroblasts. Then we go to the screening. In the virtual reality, you have the screening is represented by these three cubes. So you have one cube that you're testing molecules in order to find something that will prevent fibroblast activation. You have one cube where you want to induce cardiomyocyte proliferation, and one cube where you want to induce endothelial cell tube formation. In detail, because the video was very fast, you can see it here. So the fibroblasts are very red, the myofibroblasts are activated. you were trying to find a molecule that will reduce the activation and make them more green than red. For the cardiomyocytes, you have cells that are not proliferating, and then you're trying to find a molecule that will make the nuclei go from blue to red, which is a signal that they're proliferating. And then on the third aspect of our model is the endothelial cell tube formation, where you have the single cells in this case, and you're trying to find a molecule that will cause them to form tubes. So once we have our screening set up, we can't just press the button on the computer. We have to go to this machine, which is a liquid handler, and we have plates with 384 little wells, and in each tiny little pocket in each well, you have a molecule, and you add your cells. So then you can see one single molecule, what effect does it have on my cells? Then we move on to the other machine, which is the microscope, and you acquire the images. So you take photos of each single little well, you take 9 images of each well. So you have images of lots and lots of cells. Then you come to the computer and you analyze the images to identify possible therapeutic molecules. Okay, but this screening we performed with 1,200 drugs, but there is a lot more than 1,200 chemical compounds in the world. In fact, there's millions. So this is when we moved to the collaboration with Sasha in the Joseph Stefan Institute in order to do a virtual screen to find additional hits. So we have our data that we produced in our actual cells. Now, then using the data that we produced in the wet lab, so with real cells and using real compounds, they build a model in order to see how do these compounds react on our cells. And then we use a database from online using millions of compounds, and we add it onto this model and see hypothetically how would these other drugs react on our cells. And so then we have our virtual screening results. We have the results of what hypothetically would happen with these millions of compounds based on what happened with the 1,200 that we looked at physically. Once we have the virtual results, we go back into the real cells and we apply them. So in this case, we found one where if you have cells that you isolate directly, the cells produce a little bit of green collagen, a little bit of red alpha SMA. You activate them with TGF-beta and they become myofibroblasts, which are very green and very red. Then you add our antifibrotic drug and you can see that the intensity has reduced. So we were able to find, with the help of Sasha, a drug that is able to decrease the activation of the fibroblasts. And this can hopefully then be, this identified compound can then be developed for clinical application. So again, here in the virtual reality, you saw 3 syringes, one for blood vessel formation, one for degradation of fibrotic tissue, and one for proliferation of heart cells, which are then injected into the patient's heart to form vessels and to reduce the scar and increase the beating of the heart and heal the heart. So that's a little bit of the science behind the virtual reality screen, and thank you for your attention.
Thank you very much, Nadia. As you can understand, actually, our interest and the major topics that drove this project is cardiovascular science, because in SUGB, we have a long-lasting interest in cardiac angiogenesis and possibly regeneration. And during the last years, we had started an intense collaboration with the cardiologists, of course, here in Trieste. Initially, we thought at testing candidate therapeutic molecules, because this is the way research was moving forward 10 or 15 years ago. You just look at possible candidates that could have a therapeutic effect. Now, also in medicine, science is moving toward analysis of big data, and this is why we decided to invite to this Unconference Andrea Di Lenarda, who is the director of the Cardiovascular Center of the Azienda Sanitaria Universitaria Giuliano Isontina. He is also a friend. And over the last years, he has started some works on the use of big data in cardiovascular science. Grazie Andrea per aver accettato, and the floor is yours.
Grazie a te. Good morning or good afternoon.
But you have to unmute your microphone.
I think
it was my problem then, sorry.
It's okay?
Yes,
Can
it's
you
okay.
hear me? Yeah.
Go ahead.
Yes.
Okay, good morning, good afternoon to all of you. Yes, I am a simple, poor cardiologist with some knowledge of clinical issues and really little knowledge of your technical and deep studies that you presented now, and my compliments for your work that you finished in this Interreg. I move to my presentation. Can you see it?
Not yet.
I went to...
You should click share screen.
Okay. Sorry. Share.
Okay, great.
It's okay?
Yes.
So we are working at different level, obviously from clinical point of view at this time. This is a group of cardiologists, epidemiologists, scientists that are working together with the Minister of Health in Italian Minister of Health. I am a part of this group, not of this paper in particular, but I am part of this group since for some years. And we are trying with some critical issues about sensitive data, privacy, and so on, to merge what we have at a national level to to obtain some prognostic model to identify subgroups of people that may have different health need, just to help the health system at a national, regional level to use in the best way the resources that we have. Obviously, when we analyze this billion of data at a national level, we lose some information. We have just administrative data, and it's difficult to go in depth in subgroup of great interest in our work, that is elderly, that are usually multimorbid, and cardiovascular patient, or in particular the worst disease that we have in cardiovascular world, that is heart failure. So what we are trying in Friuli Venezia Giulia region is what we explained recently in an editorial in International Journal of Cardiology, that is to merge, to share our e-chart that since 2001 we implemented in our region. Sorry. Okay, sorry. To share what we implemented in the last 20 years. So the e-chart in which we code diagnostic items, laboratory exam, medical treatment, to better define the characteristics of our patient, and these findings are merged with the data, regional data warehouse that you see on the left, on the right, in which we have administrative data from the discharge from hospital, the general public laboratory, the public drug distribution system, and then we obtain a higher level, a higher depth of information that I shortly present with 3 examples that I collected for you. The first one is the possibility to access to our public laboratory analysis. This is just a simple example of LDL, so the worst part of lipid profile. The red bar are the proportion of patient of Friuli Venezia Giulia. As you can see, we have here 500, more than 500,000 patients of this small region. The number of very high risk people, so people that have the highest probability of cardiovascular mortality in the next 10 years are about 100,000. And you see that most of these patients are over the target of LDL that we should reach. So this is a clear example that shows that the quality of our care is till now not sufficient to control this major cardiovascular risk. And so this is the base of initiative medicine to call the worst part of this subgroup and to have a personalized treatment and personalized care to improve their prognosis. Obviously, this process is quite difficult in Italy, in the world, but in Italy in particular, because these obviously are anonymous data, and we obviously could know which patients are in this part, but this is a difficult process that we are discussing with our institution. The same is about treatment. A patient with the highest risk of cardiovascular death are treated with statin, one of the best options that we have, in about 40% of cases, and in 60% of cases, with different reasons, patients are not taking this drug. Obviously, this is the word. It's not a randomized clinical trial. It's not a registry. It is the word. For example, within this 100,000 of patients, there are almost 10,000 of patients that are not from Friuli, immigrants, etc. So with a lot of difficulties to be cared by our system. The second example is what I did in the last years in collaboration with AIFA, the Italian Agency of Drug. And this is the comparison between the Italian registry of use of anticoagulants, 1 million, almost 1 million of people, in comparison with Italian, one of the most important Italian registry, BRIS AF, of my Association of Italian Cardiologists, in which we showed that this is was published a few months ago. I showed that we are treating the oldest population of patients in the world, with the mean age of 80 years old in female and 77 years old in male. So this is a laboratory to use anticoagulants in a specific population of patients, very elderly, in which, and you see by the red box, that about 240,000 patients in Italy do not have, or at least we do not know if they have, from our data warehouse, if they have the reasons, the correct reason to use low dose of anticoagulants. So this is a big issue for our Minister of Health. We are discussing with the National Institute of Health with IFA and Minister of Health to have the authorization to go in depth in these 240,000 patients to understand if they are risking something like thromboembolic events, of if they have if they are taking the correct dose of this drug. The last example that I show to you is about heart failure. In this case, we have the maximal depth of data, of specific data, because we can merge our chart with the general regional, in this case, data warehouse. Why this is really important? When we analyze, we are cardiologists, we have a lot of registry in Trieste, in the region, in Italy, in Europe, in which cardiologists are collecting data from its population, of their population, that is a selective population of patients not too much elderly, and different from all the other cases that are admitted to internal medicine ward, that are the majority. So it's very important to look not just at the cardiology registry, but also to real world data. This is an example. This is on the left, the AMCO, National Cardiology Association registry, in which actual mortality at 1 year is less than 5%. These are the data in Friuli Venezia Giulia region of patients with heart failure less than 70 years old, all the heart failure patients, in which the mortality is 10% at 1 year, and is 30% at 1 year in patients older than 70 years old, that are the great majority of patients with heart failure. So looking only at a randomized clinical trial and registry is just one small part, not the majority of patients, of the real world cases. Another point is that half of patients with heart failure are admitted to the hospital, not for heart failure, but for other comorbidities. So we should pay attention to other issues, not just heart failure, for the best care of our patient. And this from our registry that we published 2 years ago. As you can see, patients with heart failure in real world, not in randomized clinical trial, has in at least 50%, 2, 3 or more comorbidities. So the care of this patient is much different from what the clinical guidelines are saying. And we are studying and we are writing the clinical guidelines, for example, the European Society of Cardiology guidelines. But in the real world, we are following, we are treating completely different patients that need different guidelines or meta guidelines, in which are present at the same time different diseases that concur to diagnosis, treatment, and prognosis. Obviously, prognosis, as you can see in the randomized clinical trial, we work on this type of curve, but in the real world, we are working on this type of curve. That is, with a mortality is 2, 3, 4 times higher than the selected patient enrolled to randomized clinical trial. And I think is the last or almost the last slide. Also treatment. If you can see on the right, European guidelines tell us that all patients with heart failure should be treated by 3 drugs, but in the real world, just a minority of patients are treated with 3 drugs, and the majority of these patients are treated with a low dose of evidence-based drugs. So real world and big data to analyze the real world has a great potential to understand the other side of the moon that is completely different, but is the majority of population with respect to the randomized clinical trial. The last point is just one sentence, is that we started to work, also our group started to work on machine learning and deep learning, because from this billion of data, using non-linear models, taking account interaction association between variables and drugs, and great numbers of events, we could improve our prediction of prognosis and use also what is very important for us, the 100,000 of images, ECG, electrocardiogram, echocardiogram, that I think will much improve our capacity to analyze our population, stratify our population, identify subgroup of cardiovascular patients that need much more resources to be cared. I thank you very much for your attention.
Thank you very much, Andrea. It has been very interesting, and it's a clear three clear examples in how actually different competencies and expertise will have more and more to be merged in order to deal with medicine that is becoming more and more effective, but it has to take into account much more complex situations than before, possibly also taking in consideration drug interactions and so on. So I think we could start a debate exclusively starting from this presentation. Maybe we will have time during the discussion for some concepts, but we should move on with the last part of this technical session, in which we are going to present some initiatives and synergies that could emerge from TRAIN. These are activities that were not initially foreseen by the project, but that, as science normally happens, there are more opportunities and more questions that comes during the course of experiments and activities. So I will leave the floor to Francesco Menegoni, who is a biomedical engineer. He also a lot of experience as a manager, currently serves as the head of business development of BioValley, and this is why he is a central part of the future of trains and related activities. So thank you, Francesco. I leave you the floor and I mute myself.
Thank you, Serena. Good afternoon to everybody. I will try to talk about synergies with the other projects, as Serena said, because if you look at the aim of the TRAIN project, we can see that is to increase the cooperation between the key biomedical subjects, such as research institutes, universities, and companies. And the aim for this is for increasing the innovation, the competitiveness of the biomedical research sector. Starting from that and starting from our environment, we know very well that we are today here in Zoom conference and not in real person because there was the COVID-19. And this is something that happened right now. And I will start by showing you how TRAIN and the cooperation with ICGB was a key point for being able to accelerate our processes. And the other thing that we show very well in the last presentation and the presentation before, and it was even stated in Nature journal in 2019 at the end of the year, is that today, as we see, artificial intelligence, machine learning is having a very great impact on digital health for changing medicine, drug discovery, and so on. So the first part is to show you two cases about how TRAIN accelerated other projects for us in COVID-19, for example. The first one is that together with GEB and does was to test the rapid serological test for COVID-19. What we did, and I suggest you just to look at the dates. On 13 of March, ICGB isolated the COVID-19 and had a biobank of it. Quite 7 days later, we started to analyze rapid tests as suggested by Sheridan, again on Nature magazine, and those suggested by Foundation for Innovative New Diagnostics. That is a foundation in Switzerland. And after 9 days, we compared the performance of different producers, and then we were able to have 200 tests from 2 top European producers. And what we did in 1 month, I think, from the beginning, so from the beginning from mid of March to the mid of April, we were able to validate those tests. And the results that ICZB performed were very, very interesting, because the sensitivity of those tests, if taken after 7 days of onset of the COVID, was very, very high, and even the specificity was very, very high. And so now we are validating the rapid tests on 500 hospital workers here in Trieste. And this was something that we were able to accomplish in so much short time, because we already collaborate with ICZB, and so it was very easy to collaborate to other projects. And the second case is again about the COVID-19. In this case, we provided an antiviral drug to ICZB, and we tested it on COVID in vitro. Again, looking at the dates, we started in March. The idea was of using this drug on COVID. 6 days later, the drug was inside ICZB, and in very few time, the paper was in preview on bioRxiv. You can access it here. And the result was very promising. Here I have a graph provided by ICZB that shows how the drug works very well, even in low concentration. So with something that can be used even in human. And then that's why we are now going to test it in clinical environment. So I think that train in this case was for us very important for accelerating. But there's something else that I think everybody that was here during the presentation and the 3D presentation of machine learning was fascinated by it. And I think that train inspired us even a new project. A project that is again, not only presented in Nature journals, is a project that is inside the European strategy for the digital from 2020-2030, is about the high performance computing. The white paper on artificial intelligence of the European Union talk about the importance of high performance computing. And so in this case, we started again a project that involved research and companies, as the aim of the TRAIN project is, for building the first step in having a high performance computer based on European hardware technology, and that should be available not only for research, but also for the private sector, so for the private companies. And the idea was born in January of this year. We have a preliminary location of the place where it could be located. In March, the first node has been ordered and was done with Eurotech, a European company. And in July, we will see it ready to be operative and running. So for summarizing my very short presentation, TRAIN give us the opportunity to have synergies with other projects. So to increase cooperation within those subjects and for create innovation on 3 different, in this case, cases that I showed you. So on rapid tests on COVID-19, on antiviral drug for COVID-19 and from the HPC for machine learning and artificial intelligence in health. Thank you so much.
Thank you, Francesco. I think we can stay on high performance computing with the next talk. I'm very happy to host Vincent Torre, who is a professor of physiology at SISA, always here in Trieste, which is an example of how different scientific institutions in the same region can actually cooperate and tackle the problem from different angles. He has a very interdisciplinary profile, having built a whole career in between physics and biology. So I'm very curious to listen to his talk. Thank you, Vincent.
Can you see? No?
Yes, we can see.
Okay. Now, so this project is called Virtual Biopsy and originates from a conversation I had a year ago with Miran Skrap. Miran Skrap, it's a neurosurgeon, previously was the neurosurgeon in Udine, and now he moved to Milan. And in this conversation, he said, even though I consider myself a very good expert neurosurgeon, it will be nice if I could get some help from artificial intelligence when I have to take a very important decision on what to do with a patient. And I thought that this was a very good idea. And we assembled a team, and the team is composed by Miran Skrap and some collaborators from SISA and Genoa University. And these two places are expert in machine learning and data analysis. And then we have two small companies of the region, Glance Vision Technology, who is in area di ricerca, and DataMind, who is working in Udine. Now, I will briefly explain the problem. The problem is how to deal with brain cancers and whether we can obtain we can do better by using tools from artificial intelligence and machine learning. And so this is the starting point. And Miran Skrap says that tumor pathology are very important, and you will agree on that. And this is an extremely important topic. So it's very important to have an early diagnosis, and usually there are several kinds of grades of brain cancers, low-grade cancers and high-grade cancers. And usually, if you have a high-grade cancers or glioblastoma, there is very little which can be done. And with low-grade glioma, it is possible to treat patients and provide them some good time. And it is possible not really to provide a perfect operation, but you can prolong the life expectancy. In doing this, there are some important decisions that the neurosurgeon must take, and certainly an important question is the extent of resection, whether to remove a lot of tissue, a lot of the cancerous tissue, or not so much. And so this is an important question which the neurosurgeon takes on his previous experience and on his emotional status. At the same time, there are lots of data which coming from the genomics, from biology, which can imply whether the outcome of the patient will be. And these tests are usually done by doing a biopsy, which means taking a piece of brain of the patient. And one of the main issues that Miran asked us whether it is possible to use machine learning, artificial intelligence, so that we can obtain the same information only from the MR images, magnetic resonance images, which means not to remove a piece of tissue from the patient. And if we are able to do this, this will be certainly a big advantage. Now, this is a slide which says clearly that the survival of the patient very much depends on the outcome of this biological and genomic tests. So this is an important issue. Now, let's say the key question of neurosurgery is, well, the first one is quite dramatic. Is the patient has to be operated? How much brain has to be removed? And can we avoid the real biopsy using artificial intelligence? And certainly it will be nice if we can make also some other suggestions to the neurosurgeon. And the reason is that I think that the previous speaker very properly mentioned this issue of nature, in which there is this kind of new application of artificial intelligence to understand biomedical images. And here in this slide, you see several images of different sort and origin. And the aim is to try to understand almost automatically this kind of images. And although this can be seen quite presumptuous, this is a quite feasible procedure. And the main, now in this slide, I will just summarize what is the main view of using artificial intelligence. And it goes first from a learning stage in which we try to understand these images on the basis of what has been known. So therefore, we have the large databases of images of healthy patients or of sick patients and so on. And so we have a large database of different kinds of images, and we have also the clinical outcome. And so we know that this image signals a good prognosis or a bad prognosis. And then we can make predictions, and so this is the idea. And this can be done by using different mathematical tools. And here you have some examples. We can use neural networks and different kind of methods and tools, which we are expert and we are happy to collaborate. Now, I will now show you what we have been done now. At the moment, Miran Scrap has provided us with data from about 300 patients. And so we have the magnetic resonance scans of the patient in different forms before and after the operation. And we can manage, we can organize the data. The data is different nature. So we have images, we have clinical judgment, we have also a different kind of, and this from the point of view of data processing is quite interesting. Then we are starting to do this integrated virtual biopsy, and then we have several other tools to visualization. And this is now we are able to obtain an automatic segmentation of the extent of the tumor and of the lesion of the edema. This is something that we are doing, we have already completed. And then we have started to do this, to set the basis of this virtual biopsy. And here, this is so this is a movie, and this is just a summary to show you what we have been doing. And so there is a first section here. We are dealing with the data from the patient. We have the name, and then we can project on a standard map of the cortex, the extent of the lesion of this patient. And then we can do some sort of statistics. We can forecast what is the survival of the patient given different genetic tools, the extent of the resection and so on. And so this is something that is already working. We have to improve and we have to obtain better data. So what will be very nice to provide is to not only give some sort of a way in which the surgeon, neurosurgeon can look and explore the available data. Will be nice if we can provide the best extent of removal, which is called the EOR. Will be nice to try to suggest to the surgeon the best combination of radiotherapy and chemotherapy, and also the best rehabilitation therapy. These things are based on an intelligent exploration and navigation of similar cases well known in the literature. Therefore, the system will be quite effective and useful if we are able to gather lots of data, lots of clinical history of patients, and with a nice set of data, we expect to be able to provide a good tool to clinicians, surgeons, and also to have some product coming from the region, which can be useful also from an industrial point of view. Thank you for your attention. Thank you.
Thank you very much, Vincent, for this very nice and futuristic presentation. And with this, I think we can conclude the technical session and move toward the last session, which is about policymaker. This is an Interreg project, so it's not just technical details in science, but it is also creating a network of good practices and policies that could be somehow combined, transported, and arranged together. And for some of these presentations, there will be a translation that has been requested to ensure full understanding of contents. We did not think at having a translation for all the speakers, otherwise in a webinar it would be too heavy to listen to, particularly for the Unconference format, since we didn't know exactly what speakers had to say. But of course, if anybody is willing to have a translation of the presentations of this event, you just mail us and we can provide everything very soon. So first speaker of this policymaker session is Luciana Zanier, who has been a key person in fostering the dialogue between science and politics here in our region. She has a great experience in the European Commission and recently joined some regional activity. So thank you, Luciana, for being with us, and I leave you the floor.
Can you hear me now?
Yes, we can hear you.
Okay. And see the presentation as well?
Yes.
Okay. So first of all, let me congratulate with all the partners, because I'm very impressed by the results. And thank you for inviting me to take part to this Unconference. It's my first Unconference. So I'm very excited. Specified in the agenda and introduced by Serena, this is the policymaker session. And so we should discuss about how the research proposed in TRAIN project can have an impact on the health system in the program area. Well, in my brief presentation, I would like to touch a specific aspect of the relation of this relation between research and science and its impact on policy. As mentioned by Serena, I became familiar with this special role of science and policy interface in a previous working experience. I worked for several years at the European Commission at the Joint Research Center. That is the science and knowledge service of the European Commission. So it's the service that whose mission is to support the European policies with evidence. And I found very similar characteristics at ICGB, because ICGB is an intergovernmental international organization. So the members are the governments of the 65 members, but whose main mission of the institution is research. So in ICGB, there is a coexistence of scientists, researchers, and policymakers. So we all know that policymakers are increasingly confronted with a wide range of complex and interdependent problems. We see now health, for example, that is, I mean, the crucial problem nowadays, but climate change, economic inequality, aging population, water scarcity, all these kind of very important problems. And the policymakers turn to science for a better understanding of the reason and the mechanisms, and to identify the possible solutions. So they need their decisions to be informed by the best available science. This is crucial because the wrong policy can result in critical economic and social cost and destroy the trust in governing institutions. So the good news is that never before in human history has so much scientific knowledge been produced and is never been so easily accessible. We now have a better understanding of our planet, of our economy, of our society, of ourselves than any other time in history. But the process of translating the scientific knowledge into policy relevant evidence, this is not simple. So, as in the slide, the words of science and policy can be seen as separate and in collision, having different perspectives. They have different goals. The aim of science is to know. The goal of politics is to act. So typically, it's a lack of mutual understanding and empathy between the scientific and political community. It is a very complicated relation. So since we have here either scientists or policymakers, we can joke a bit. And we can illustrate the different perspectives. I cannot go on. Oh, yeah. So with this, the analogy in this slide. So the latitude and longitude given is for Trieste. So it's this image that this man on the balloon, can you tell me where I am lost? And the other man say you are at latitude 45.6495 north and longitude 1377 east and so on. I am standing at 300 meters above sea level, so you must be at about 330 meters. Oh, sorry. And so, no, the man on the balloon say, you must be a scientist. I ask you a simple question and you give me too much information and I am still lost. And the other replies, you must be a policymaker. You come out of nowhere with your questions. I give you the most accurate and precise answer I can. You are still lost and you blame me. So okay, this is a joke, but I mean, sometimes the dialogue is very, very difficult. So we know we need scientific evidence to inform policy, and we now need to learn how to do this better. It is extremely important bridging these two distinct worlds of science and policy to increase the impact, and this is the important thing, to increase the impact of research evidence on policy making. The science policy interface is a very specific field with its own culture. It is complex with large potential for misunderstanding and missed opportunities. Science communication happens in a complex context whose elements include the content to be communicated, the communicator, the audience, the channels of communication. And there are also many other sources from which policymakers, but also users, citizens, business may obtain additional and perhaps conflicting or inaccurate information about science. So it's important, therefore, to promote a framework to facilitate this dialogue in order to provide more effective scientific evidence to inform policy. And this approach can be based on 4 elements. So trust, this is the most important, because if there is no trust, the evidence will be ignored. Trust is indispensable to have a real impact on policy making. We need trustful relations between scientists and policymakers. If there is no trust, there is no acceptance. And if there is no acceptance, there is no mutual interaction. So building trust and confidence in our knowledge producing institution is crucial to create the appropriate conditions for a positive general attitude towards knowledge implementation. Timing, this is vital. Scientific evidence should be provided as early as possible in the policy cycle, before fundamental policy positions are taken, and it should ideally frame the discussion. So it should not be provided at the end of the process, at the end of the policy cycle, when it's probably too late to influence anything. And there is also a specific case that is nowadays evident. It is the case of the scientific support to crisis, when speed of response is crucial. So then we have other 2 aspects, the form. So the supply and demand for scientific evidence will be best served by bridging policy and science as close as possible. Scientists need to be an accepted part of the policy process, of the policy cycle, consulted at all stages of the policy cycle, from the initial discussion about potential new policy throughout the ex post assessment of the policy impact. So the relation has to be interactive. This means making sure that scientists participate formally or informally throughout the policy cycle, and that policymakers are continuously involved in knowledge creation and management. Because the closer involvement of policymakers in the knowledge process will increase the likelihood of knowledge having an impact on policy. And then format, also this is relevant, because policymakers need brief, short, with visual inputs, so that they can quickly understand the main message, and that they do not have to dig in an extensive, highly technical report. Especially in my previous job, I saw these reports of 60 pages. It's impossible that a commissioner of the European Union reads 60 pages for taking a decision. So these are the four elements, in my opinion. I would like to close my presentation with a quote of a very important biologist. As pointed out by Edward Osborne Wilson in his really inspiring book, Consilience, we have no lack of knowledge. The downside is that this creates a confusing place for policymakers. The many sources of evidence means that the policymakers are now facing a problem of abundance, not scarcity of knowledge. We need to add knowledge management and sense making. We need tools and competence to be able to collect and collate all this huge amount of scientific knowledge that is available, to quality check, to process the data and knowledge, and to select only the policy relevant part for the onward transmission to policymakers. So target evidence is the key to successful scientific support to policy. So thank you very much for your attention and for inviting me.
Thank you, Luciana. Very
I'll try now to stop sharing.
Inspiring, and I think good thought for further discussion and follow up. So we move on and we move to the next speaker, who is Professor Antonello Pezzini, who is EESC Counselor, so consigliere del Comitato Economico e Sociale Europeo, who will give us his perspective on how these kind of Interreg projects might have relevance also at a wider perspective. Thank you very much for being with us. I think that he will speak in Italian and there will be a translation.
Pronto? Si sente? Buongiorno. Sì. Bene, grazie.
We hear you.
Grazie. L'intervento sarà sintetico in 5 punti. Primo punto, grande utilità dell'uso dell'intelligenza artificiale in questo progetto. Si è fatto riferimento al libro bianco della Commissione. Aggiungo che il libro bianco vuole che l'intelligenza artificiale sia antropocentrica, cioè sia rivolto all'uomo, come in effetti sta avvenendo con il lavoro che voi state facendo. Traduce?
Can you hear me all right? Can you hear me? Okay, so thank you very much for your invitation. My presentation will be extremely brief. I will talk about 5 points. The first is the usefulness of artificial intelligence in the project that you have illustrated today. And you also mentioned, you made a reference to the white paper of the Commission. And according to the white paper, artificial intelligence should be man-centered, and this is what is happening with your project.
Secondo aspetto, entriamo nell'industria 4.0. È nel progetto ed è stato citato anche dagli interventi precedenti, la grande mole di dati, big data. Attraverso questa grande mole di dati, attraverso l'intelligenza artificiale, noi ci possiamo proiettare verso il futuro.
The second point regards Industry 4.0, and it is present in your project as well, as you mentioned before. Another crucial aspect is the big amount of data, which, along with artificial intelligence, can project us towards the future.
Per il futuro intendo il foresight. Il foresight è uno degli elementi anche fondamentali della DG Innovation, che si basa sul principio che il futuro non appartiene alla sfera di cristallo, ma il futuro è il risultato del lavoro degli uomini.
And by future, I mean exactly foresight. Foresight is a concept also included by the DG Innovation, General GD Innovation. And future means, foresight means that future is built by man. It does not depend on a crystal ball.
C'è un'analogia tra il futuro della storia e il futuro della tecnologia e della medicina. Per la storia, Edward Cara ci insegna che se vogliamo cercare di capire il futuro, dobbiamo studiare a fondo il passato e il presente per avere gli elementi di interpretazione del futuro.
And there is an analogy between the future and the history and technology and medicine. According to Edward Cara, we cannot understand the future if we don't study the past and the present. Those are the keys to understand the future.
Nel campo della medicina e della tecnologia il discorso è analogo. Noi dobbiamo studiare attraverso degli algoritmi, attraverso i big data, una serie di elementi fondamentali, fare una comparazione tra questi elementi e da questi trarre alcuni insegnamenti per ciò che si verificherà in futuro. È la stessa cosa e voi lo state facendo con questo lavoro di Interreg.
Yes, and for medicine and technology, the principle is the same. Through big data and algorithms, you have to understand, to evaluate fundamental elements and compare them. And through this comparison, you can understand and create the future. And this is exactly what you're doing with your project, Interreg project.
Il quarto aspetto è la trasferibilità del processo. Tutto quello che state studiando, soprattutto per la previsione delle malattie, della biotecnologia, della medicina, che è fondamentale, può essere trasferita nella tecnologia. La pandemia ci ha dimostrato che anche un'interruzione di servizio in una scala mobile, in un ascensore, in un ospedale o in una clinica provocava un grande disastro. Noi dobbiamo prevenire questi fatti attraverso lo studio dei fenomeni ed evitare che succedano.
Yeah, the fourth point is the transferability of the process. You need to be able to predict a disease in biotechnology, in medicine. This is exactly what technology is doing right now. And for example, the pandemic has taught us all this. For example, if you have an escalator, if you're working in a hospital, a power shortage is really crucial. So you need to predict that and prevent that. That is the same thing that you can do in medicine as well.
Quinto e ultimo aspetto, l'articolo 168 del Trattato di Lisbona e l'articolo 35 della Carta dei diritti fondamentali, che fa parte del Trattato di Lisbona, ci dicono che noi dobbiamo mettere il concetto della salute in tutte le politiche che dobbiamo fare in Europa. Per questo dobbiamo fare, e ce lo dice l'articolo 168, tutta una grande raccolta di dati attraverso la trasformazione digitale e dobbiamo avere una grande sorveglianza.
The fifth point regards the Article 178 of the Lisbon Treaty, as well as the Article 35 of the Charter of Fundamental Rights, which is included in the same treaty. According to those articles, health should be included in all the policies of the European Union. And that is done through the collection of a huge amount of data and digital transformation, as well as surveillance.
Per questo, agli iniziali 2 miliardi di euro che erano stati stanziati, previsti nel programma 21-27 della Commissione europea, grazie al nuovo programma RESC-EU, RESC per l'Europa, che è inserito nel Next Generation, sono stati appostati ancora 8 miliardi 451 milioni, quindi un totale di 10 miliardi di euro che saranno disponibili per continuare in questi lavori che voi avete iniziato.
And for this reason, 2 billion euro were allocated in the 2021-2027 program of the European Commission. And now through the RESC-EU program and Next Generation, other 8.451 billion euro, so more or less a total of 10 billion, will be available for further projects like the one that you are carrying out today.
Questo voi lo potete trovare nella proposta di regolamento che stiamo discutendo proprio in questi giorni a Bruxelles, che è il COM 2020-405 del 28 maggio 2020, è uscito pochi giorni fa. COM 2020-405. Trovate queste indicazioni anche di appostazione di bilancio. Grazie.
And all this is contained in the regulation proposal COM 405 of 28th of May 2020, so it's really recent, and you can find all of these in that proposal. So it's COM 2020 405, and please have a look at that. You will find all the allocations regarding these projects. Thank you.
Buon lavoro e complimenti.
So good luck and congratulations on your project.
Grazie professor Pezzini, è molto incoraggiante sapere che le nostre attività sono state ben recepite e che c'è l'interesse di continuare a investire in progetti in dimensioni del genere. So we are all happy to hear that actually our activities have been well received, and it is very encouraging to hear that the European Commission is going to further invest in these kind of initiatives and programs. And with this, I think we can move on to the last speaker, Niccolò Querci, who is a policy assistant at the European Commission for the DG Research and Innovation. So we are still within the European Commission and looking forward to your speech. Thank you very much.
Thank you. Good afternoon to everybody. I hope you can hear me well. Now I will try to share with you my PowerPoint presentation. Yes, today we present you Horizon Europe, the new framework program for research and innovation for the period 2021-2027. I will talk about the structure, the main novelties, and the added value in promoting cooperation and increasing innovation. I have 10 minutes, so I cannot go too much in details, but I'm fully available after this session or also after the end of the conference to reply to your answer also in a written way. To address the challenges Europe is currently facing, the European Union needs to invest more in research and innovation. Union-funded research and innovation activities produce demonstrable benefits compared to national and regional R&I support. Action at union level enable transnational cooperation and worldwide competition to ensure that the best proposals are selected. This, of course, also raises the level of excellence, and it provides visibility for leading R&I. This also supports transnational mobility and attract the best talent. A union level program is best placed to take on high risk and long-term R&I project, and it can also generate a breadth of scope and economy of scale that could not otherwise be achieved. Horizon Europe will be built on the achievements and the success of the current research and innovation program, Horizon 2020. So Horizon Europe will keep the three-pillar structure, will be based on excellence and maintain the test funding rules and procedure of its predecessor, Horizon 2020. Innovation always starts with excellent research. So the program under Pillar 1 will continue to drive scientific excellence through the European Research Council and the Marie Skłodowska-Curie Fellowship and Exchanges. The second pillar focuses on global challenges in the European industrial competitiveness. It's designed to fund research and innovation, tackling societal challenges and reinforce technological and industrial capacities. This pillar contains its clusters that address the full spectrum of global challenges you can see on the slide. The integration in cluster is designed to incentivize a systemic approach with cross-disciplinary, cross-sectorial, cross-policy, and international cooperation. In this pillar, you can also find one of the most important novelty of Horizon Europe, the research and innovation missions. And here you can also find the industrial partnership. This pillar will benefit from the scientific advice, technical support of the Joint Research Center with the Commission Science and Knowledge Service. Finally, the third pillar, Innovative Europe, will help the European Union to become the front runner in market creating innovation. Here you can find two main instruments, the European Innovation Council and the European Institute of Innovation and Technology. As I already said, both us, the Commission, and the final users, so the stakeholders, are quite satisfied about Horizon 2020. However, from the Horizon 2020 interim evaluation, we saw there is still a room for improvement. We learned some lessons. And these lessons are that we need to support more breakthrough innovation. We need to create more impact through a mission orientation approach and citizen involvement. We need to strengthen international cooperation, to reinforce openness, and to rationalize the funding landscape in order to encourage participation. In order to support the breakthrough innovation, Horizon Europe will introduce the European Innovation Council. The European Innovation Council, for which a pilot phase has already started under Horizon 2020, it will be a one-stop shop to bring a disruptive and breakthrough innovation from lab to market application and to help startups and SMEs to scale up their ideas. It will provide direct support to innovators through two main funding instruments, one for the early stages, the Pathfinder, and the other, the Accelerator for development and market deployment. With the European Innovation Council, we are aiming to do for innovators what we are already doing for top researchers. So, for example, the EIC will focus on individual entrepreneurs and companies, incentivize bottom-up ideas, keeping procedures simple. The European Innovation Council will significantly simplify and streamline the current support and fill any gap between the grant funding in the other part of the program and other financial instruments. In order to create more impact through mission orientation and citizen involvement, Horizon Europe will introduce the concept of missions. Horizon Europe mission will have some common characteristics. They will have a clear EU added value. They will be bold and inspirational. They will indicate a clear direction, and they will be targeted, measurable, and time-bound. They will be centered on ambitious but realistic research and innovation activities. They will spark activities across disciplines, sectors, and actors. And finally, they will be open to multiple bottom-up solutions. The 5 mission areas that we already identified together with the co-legislators, so the Council and the European Parliament, are cancer, soil, health and food, climate neutral and smart cities, healthy oceans, and adaptation to climate change. Another aspect that could also be important for you is the synergies with the other union programs. In developing the next generation of union programs, the Commission has made a real step change in promoting synergies. Close attention has been paid to enabling synergies from the design stage through, for example, mirroring provision in the respective proposal. Horizon Europe will promote effective and operational synergies with our EU programs and policy in order to encourage faster dissemination at national and regional level, in order to uptake research and innovation results, in order to enable the pursuit of common objectives and common areas for activities. For example, maybe someone of you already know the Seal of Excellence. The Seal of Excellence will allow other funders, notably managing authorities involved in the European Regional Development Fund or in the European Social Fund, to benefit from the Horizon Europe proposal evaluation. Furthermore, with regard to the implementation, the harmonization with other EU programs will be enhanced. Where are we now? The Commission services are now co-creating the first draft of the strategic plan of Horizon Europe. We will launch soon a short web-based co-design exercise, probably in September, during the Research and Innovation Days that will be held online, not anymore in Brussels, from the 22 to the 24 of September. This process will help us in the implementation phase by improving how we present the link between R&I investment and EU policy. And it will also help us to ensure the widest possible ownership of the plan, and will facilitate synergies with other EU and national program initiatives. So our plan is to have everything ready by the end of the year. So to have the first work program for Horizon Europe, work program that will cover the first 2 years, in order to launch the first call. Please note that the process that I have described in these slides is contingent on an agreement on the MFF. The original proposal of the Commission was to assign 100 billion euro to Horizon Europe. On the 27th of May, the Commission, as already Mr. Pezzini reminded, put forward its proposal for an ambitious recovery plan in response to the coronavirus pandemic. So the Commission created a new recovery instrument, Next Generation EU, to respond to the massive investment needs and to ensure that recovery is even, inclusive and fair for all the member states. Through this instrument, a 13.5 billion increase of the Horizon Europe budget is proposed. This budget increase will be used to provide European support for health and climate-related research and innovation activities. From my side, that's all. Here you can find the website where you can find more information, and my email address if you have any questions. Thank you.
Thank you very much for this very comprehensive and novel presentation. It's always interesting to see and listen what's new and what's coming up. I think that with that, we can close this session. It has been really a nice opportunity to take into consideration different aspects of stakeholders and people that eventually have to communicate and be both the proponents and the beneficiaries of these kind of interregional programs where we need to work together to try to boost the capacity and innovative features of our territory. So I think that with that I can stop and leave the floor to Diego Bravar, who is going to summarize what TRAIN has been and what we believe TRAIN can serve as a platform for the mid-long-term future. Thank you, Diego.
I would only start with some presentation of BioVale investment, so we started 5 years ago with the idea to invest not only in Friuli Venezia Giulia region, but in Veneto, Trentino Alto Adige, Austria, Slovenia, and Croatia. The idea was started for my family and friends' offices for investing in what we call biotech technology. And biotech is a market of 600 billion euro that's growing 7% per year worldwide, and more or less Europe is 1/3 of this. And medtech or pharmaceuticals is important as capital spending that we have in Europe. For this reason, BioValley start the TRAIN project, deal with the communication, as you said, and dissemination, together with the Ljubljana Technology Park of the research activity of ECGB, Joseph Stefan Institute of Ljubljana. This dissemination was aimed primarily to over 30 companies compared to 28 of Slovenia in the biotech net operating company in the healthcare sector. The expertise company operating in environmental biotech in Venice Science and Technological Park, and biotech company established in the Ljubljana Technology Park. In particular, what has so far been developed as a digital platform model for the biotech industry could be transferred to Italian and Slovenian health services, implementing further artificial intelligence program already launched, also with the support of CISA and with other companies in the biotech network. As well as with the support of the Joseph Stefan Institute. Furthermore, BioValley Investment will acquire from Eurotech a new digitalization platform, private cloud data center, which it will install in the Trieste Convention Center of the Porto Vecchio Trieste, and which will be activated by September 2020 for ESOF, and will be available to the private company of the sector. This new technological platform will thus also be able to support the 5th edition of the Eurobiotech event, which will take place in this year during ESOP 2020. As always, both research institutions and private institutions will be involved, who will also be able to present electronically the result obtained with the European Portfolio or cross-border funding. It could be also be activated with the homologous public cloud data center, which already has been used during the TRAIN project for a cross-border digitalization and prevention system in the health sector with artificial intelligence program. To this end, it would be appropriate, in my mind, to promote by some participant of the TRAIN project the establishment of an economic European interest group in which to involve both research institution and biotech innovative company, not only in Italy and Slovenia, but also in the Central Eastern Europe and in the Balkans. This economic group of European interests could directly access, in my mind, to the EU program EU for Health, and mentioned by the speech of Professor Antonello Pezzini and by the Professor Niccolò Querci. Thank you very much.
Thank you, Diego. This is something we have discussed a lot during the last months. Of course, we will be very happy to continue TRAIN activities because we really think that having established this network of people who have really worked together during these years could be the central nucleus to be enlarged to other participants, as you said, here in Trieste, in Slovenia, but also extending to East Europe, where we all have a lot of collaborators and ongoing activities. Of course, having the possibility to present our activity in the context of ESOF would have been a tremendous advantage to liaise with other partners and start gathering, but of course we will have a future occasion, even if a train will be officially ended by the time we will be able to participate to further events. So I think that more or less we have done with this Unconference and also with the project. As mentioned, this was the last event, so I'm half happy and half sad that this adventure has officially terminated, but as said, we will keep working together because it started new activities, new thinking, new collaboration. So of course we will be a team that will still exist. If there are any questions from the audience, I will be happy to accept. I know that discussing in this format is not as nice as being alive. So if there is any, I think we can redirect the question to the relevant person. But I also think that all the speakers will be available for further discussion or additional follow-up of this event, even by email or other means that we can identify. So is there any question that we received?
No.
Ciao, I had a question for Andrea Vilenarda, Dottore. I was wondering how often the screens that we do, the drug repurposing screens, actually end up in a relevant medication that you use in the clinic. Is it something that you've seen to be a successful approach?
Yeah. Okay. So the question is how often I screen. Can you repeat the question?
Yes, of course. How often does a drug repurposing screen end up with something that is actually...
Can you repeat again, because the voice was interrupted.
Maybe I can do that. So maybe I can repeat the question.
Serena, may you repeat?
Yes, so the question was, how often a drug repurposing actually ends up with novel use of existing drugs that can be of real benefit for the patients?
I think that we have a big issue in medicine, that is that most patients are not adherent to available treatment for cardiovascular disease, for example, in the sense that I think that the main issue is to apply what we have, and we are, as I showed, we applied we perfect implemented drugs in our patient in a small percentage of cases. On the other hand, this is one point. The second point is that new drugs are very frequently proposed in scientific reports. The drugs that are really changing the natural history and prognosis of our patients are relatively few. Because we have a big background of medical treatment for each disease. Anyway, the mortality and hospitalization for each cardiovascular disease or other diseases are still very high. So I think that the room to have new drugs probably with different mechanism are quite high.
And if I can add, I mean, I'm very happy to have heard the intervention of Niccolò Querci about this Innovation Council, because I think that in the cardiovascular science, for instance, the use of biological drugs is almost null. So if you think at monoclonal antibodies, recombinant proteins, these are drugs that comes from the academic research. So big pharma actually try to slightly modify drugs that already exist and are essentially chemicals. While to develop a biological drug, the process is much more complex. Often these kind of potential drugs come out from screenings that have been done for academic research, but then to help the researcher to have the IP protection and starting a startup to have a proof of concept, and then to transform a startup in something that can be appealing for a big pharma. So trying to expand the economic power of the startup is very difficult, particularly in some parts of Europe. And I think that Italy and Slovenia are among these portion of Europe where there is a real need not really to make a startup, because for that you have some possibilities to apply. But then once you have a startup, I think that 99.9% of the startup eventually fail because there is not the possibility to make the jump and reach a trial of a certain extent that convince the big guys to acquire the company. And so I think that this is something where we really need some investment and some change in mentality to really bring some of the results that come from the academia, because this is a screening starting from scratch is something too risky for a company that cannot invest in 20 years years anymore. So it's something that comes from the academia, and then it's really difficult to translate into something useful in the clinic. So having a platform and a program that help this transition, I think would be essential to bring really new drugs to the cardiovascular field. Any other thought or concept, or anybody willing to share anything else? If not, I think we can close here this very interesting Unconference. I think we kept the format quite nice. It has been very variegated, not too boring, everybody bringing his own perspective. I thank you all the speakers, all the people who participated, also our regional contact. I saw that they were there, and we really want to express our gratitude for having had the possibility to do what we did. It is, as said, a very nice and inspirational experience also for future moves in our region. So thank you very much, everybody, and hope to see you soon in person.